Welcome. Today, we will explore the emerging role of biosynthesized copper nanoparticles in biotechnology. This is a nanomaterial with growing applications in a multitude of fields. For example, in agriculture, biological applications, in the environment, and industry sector. Copper is a transition metal that is essential to the function of very useful enzymes like lacase that is used in the transformation of wood into paper. A nanoparticle is a very small particle that has dimensions in the nanometer range, typically 1 to 100 nanometers. Because of their size and properties, nanoparticles interact with cells in distinctive ways. But Moisés, how are copper nanoparticles biosynthesized? Well, copper nanoparticles can be biosynthesized using plant extracts, for example, a pencil cactus extract, where bioactive compounds such as flavonoids, phenols, and enzymes act as a reducing and stabilizing agents, converting copper ions into nanoparticles in a sustainable process. Let's observe an example of a real-world problem where copper nanoparticles offer a promising solution. Nicotiana tabacum, or more commonly tobacco, is an important global economic market with an estimated value of 789 billion euros. Plant viral infections incur losses in the hundreds of millions of euros every year. One of the main contributors to these losses is the tobacco mosaic virus, which significantly reduces both the yields and the quality of tobacco crops. This viral infection affects tobacco plants by stunting growth, causing leaf discoloration and impairing plants' ability to produce viable tobacco. It has been demonstrated that biosynthesized copper nanoparticles with orange peels can reduce the spread of tobacco mosaic virus infections in tobacco plants. These nanoparticles can decrease the infection spread by up to 4.5 times at a concentration of 100 milligrams per liter. So let's observe another example. Nitroaromatic compounds are emerging pollutants that pose significant environmental and health risks. These compounds are widely used in industries such as explosives, pesticides, and pharmaceuticals. However, their persistence in the environment, toxicity, and bioaccumulative nature make them dangerous contaminants. Biosynthesized copper nanoparticles have emerged as a highly effective catalyst in the reduction of nitroaromatic compounds. The effectiveness of these biosynthesized copper nanoparticles is attributed to their ability to interact strongly with the nitro group in nitroaromatics. They are achieving remarkable conversion rates at 96% in just 6 minutes at 35 degrees, maintaining a high efficiency rate across at least 8 consecutive catalytic cycles. Such resilience not only enhances their cost effectiveness for large-scale industrial and environmental applications, but also reduces their environmental footprint by minimizing waste. In conclusion, biosynthesized copper nanoparticles exhibit exceptional antiviral and catalytic properties, making them a versatile tool for addressing diverse challenges. This combination of antiviral effectiveness and catalytic durability highlights their promise as green, innovative solutions for both environmental and agriculture applications. To wrap things up, here are a few final questions from what we have learned. Is copper an essential metal in living organisms? Which virus was mentioned in the video as being controlled by copper nanoparticles? At what temperature and time did copper nanoparticles achieve a 96% conversion rate in reducing nitroaromatics? Thank you for watching. I hope this has sparked your curiosity about the incredible potential of copper nanoparticles.